Welcome in to Radio-ish EBM, where questions get sharp and answers take shape, one idea at a time. Let's begin.
Imagine a doctor being able to predict how your body will react to a certain treatment based on your unique molecular makeup.
Mm-hmm.
That's the promise of proteogenomics,
Right.
a field that is like reshaping nephrology.
Yeah.
Today, we are going to do a deep dive into how proteogenomics is being used to understand, diagnose, and treat kidney diseases.
Right.
We're going to focus specifically on two diseases today.
Okay.
ccRCC, which is clear cell renal cell carcinoma, and a genetic disorder called ARPKD.
Okay, sounds good.
This is Radio EBM.
So what is proteogenomics?
Proteogenomics is about combining different levels of biological information.
Okay.
Genomics, transcriptomics, and proteomics.
So we're talking like DNA, RNA, and proteins.
Yeah, exactly.
Oh.
So we're looking at your DNA blueprint, seeing which genes are actively, creating instructions and then analyzing the actual proteins that are being produced.
So rather than just having, a basic sketch, we're getting a high-definition 3D model of what's happening inside your kidneys.
Yeah, exactly.
Wow.
Yeah.
Okay, so walk us through how this proteogenomics pipeline actually works.
Sure. So it starts with DNA sequencing,
Okay.
which allows us to pinpoint those genetic variations that can have a significant impact on your health.
Okay.
Think of it like looking for tiny glitches in your DNA code that might be causing problems.
Oh, so like a typo.
Yeah, exactly like a typo.
Okay.
Then we move on to transcriptomics.
Okay.
We use RNA sequencing to see which genes are actually switched on
Okay.
and producing those RNA transcripts.
So which genes are actively like making instructions?
Exactly.
Okay.
And finally, we get to the proteomics part,
Okay.
using techniques like mass spectrometry to identify and quantify the proteins that are actually being made.
Wow.
We can even see any modifications those proteins might have undergone.
So we're not just looking at these layers separately.
Right.
We're integrating them to get a much more complete picture.
Yeah, that's the key. By correlating the proteomic data with the genomic and transcriptomic information, we can identify brand new proteins or even variations in known proteins that are directly linked to a disease.
It's like solving a puzzle, right?
Exactly.
Each piece, the DNA, the RNA, the proteins, gives us a clue to understand the bigger picture of what's happening inside the kidneys.
Yeah.
Now, the real power of this approach is in how it can be applied to specific diseases, like ccRCC.
Right.
I know that's a type of kidney cancer.
Yeah, ccRCC originates from the renal tubular epithelial cells.
Okay.
Those hardworking cells that line the inside of your kidney tubules.
And so that's interesting because it might seem like a urology issue at first glance,
Right.
but its impact on kidney function is a major concern for nephrologists.
Exactly, yeah.
Now, I know there was a study that used proteogenomics to understand ccRCC's treatment response, and this study focused on sunitinib, which is a tyrosine kinase inhibitor.
Right.
Can you tell us a bit more about that?
Yeah, so tyrosine kinase inhibitors work by blocking specific enzymes that promote tumor growth. But the thing is, not everyone responds to sunitinib the same way.
Wow.
Some patients have great results, while others see little to no improvement.
So how did the researchers use proteogenomics to understand this variation in treatment response?
Well, they integrated proteogenomic data to compare those who responded well to sunitinib with those who didn't.
Okay.
And they found that certain chromosome alterations combined with the activity of specific signaling pathways, particularly the mTOR pathway,
Okay.
played a significant role in determining treatment response.
Can you explain what the mTOR pathway is?
Sure.
In a way that someone without a background in molecular biology might understand.
So think of the mTOR pathway as a control center inside your cells,
Okay.
regulating things like cell growth and metabolism.
Okay.
Sometimes in ccRCC cases, this pathway gets stuck in overdrive,
Okay.
like a car with a jammed accelerator, pushing the cancer cells to grow uncontrollably.
Okay.
Sunitinib can act like a brake in these cases, slowing down that uncontrolled growth.
That's a great analogy.
Thanks.
Okay, so the study found that chromosome alterations and mTOR activity were key factors in how well someone responded to sunitinib. Did they uncover anything else?
Yes. They also discovered that the tumor microenvironment, the surrounding cells and tissues, also influenced treatment response.
Okay.
But here's where it gets really exciting. Based on these findings, They were able to develop a multi-omics classifier.
Okay, a multi-omics classifier. What is that and why is it important?
It's a tool that can predict based on a patient's unique molecular profile
Okay.
whether they are likely to benefit from sunitinib treatment.
Wow.
So no more guessing games.
Yeah.
You'll know right away if the drug is likely to work,
That's incredible.
saving valuable time and resources
Yeah.
and potentially sparing you from unnecessary side effects.
This is truly personalized medicine in action,
It is.
using proteogenomics to guide treatment decisions and
Beautiful.
potentially improve outcomes for ccRCC patients. Now, I know that there's another study that delves even deeper into the molecular landscape of ccRCC.
Yes.
This one compares genomic, transcriptomic, proteomic, and even phosphoproteomic data.
Right.
So that's looking at protein modifications
Yeah,
from tumor samples and adjacent normal tissue.
exactly.
That's a lot of data.
It is.
What did they find out from all of this?
This is Radio EVM.
So they were able to identify key genomic alterations like chromosome arm level changes and
Right.
somatic mutations.
Mm-hmm.
Then they traced how these changes affected mRNA protein and phosphoprotein levels.
Okay.
They essentially saw the ripple effects of these genetic changes all the way down to the protein level.
So they were able to see the full impact of these changes,
Yeah.
starting from the genetic level, working all the way down to the protein.
Exactly.
Wow, this is fascinating.
It is.
What else did they find?
They were able to identify distinct immune-based subtypes of ccRCC,
Okay.
each characterized by specific cellular pathways and genomic alterations.
So even within ccRCC, there are different subtypes with unique molecular signatures.
Exactly.
It's so fascinating.
And these subtypes aren't just academic curiosities.
Right.
They're actually linked to different clinical outcomes and responses to therapies.
So could we use this knowledge to develop more targeted treatments?
That's precisely what researchers are hoping to achieve.
Okay.
This in-depth molecular understanding is paving the way for more personalized and hopefully more effective treatments for ccRCC.
That's really exciting.
Yes.
Now, while proteogenomics is transforming our understanding of ccRCC,
Mm-hmm.
it's also shedding light on inherited kidney diseases
Right.
like ARPKD.
Yeah.
Can you tell us a bit more about this condition?
ARPKD stands for Autosomal Recessive Polycystic Kidney Disease.
Okay.
It's caused by mutations in the PKHD1 gene, a gene crucial for normal kidney function.
Okay.
When this gene is mutated, it leads to the abnormal growth of the renal tubule epithelium,
Okay.
causing the formation of cysts, those fluid-filled sacs that disrupt the kidney's filtering process.
And how does proteogenomics help us understand the molecular mechanisms at play in ARPKD?
One of the key things it can do is identify changes in the expression of proteins encoded by the faulty PKHD1 gene.
Okay.
We can see how the genetic mutations in PKHD1 actually translate into altered protein production.
So we can see the direct link between the genetic mutation and the resulting changes in protein production.
Exactly.
That then leads to the development of the disease.
Exactly. And this knowledge is crucial for developing effective treatments.
Okay.
Proteogenomics is also helping researchers pinpoint potential therapeutic targets in ARPKD.
Okay.
Some promising research is looking at blocking epithelial growth factors,
Mm-hmm.
which are involved in that abnormal cell proliferation we talked about.
So if we can target these growth factors,
Right.
we might be able to slow down or even stop cyst formation.
That's the hope.
That's great.
Another exciting area where proteogenomics is showing promise is in using extracellular vesicles
Okay.
as a non-invasive diagnostic tool.
I remember you mentioning those extracellular vesicles earlier.
Yeah.
Remind me, what are they and how can they help us diagnose ARPKD?
Imagine them as tiny delivery trucks
Okay.
sent out by your cells.
Okay.
They carry all sorts of molecular cargo, including proteins and RNA.
Mm-hmm.
When cells are stressed, like in ARPKD,
Oh.
they release different types or amounts of these vesicles.
Okay.
So by analyzing these vesicles in a patient's urine,
Great.
we might be able to detect ARPKD early on or monitor disease progression non-invasively.
Now, zooming out a bit, how is proteogenomics impacting the search for biomarkers in kidney diseases more broadly?
That's a great question. So biomarkers are like signposts giving us clues about disease presence, progression,
I see.
or response to treatment.
Right.
They're becoming increasingly important for early diagnosis and personalized care in chronic kidney disease, or CKD.
So you mentioned large prospective CKD cohorts earlier.
Yep.
How are these large-scale studies aiding in biomarker discovery?
These studies are incredibly valuable. They follow patients over time,
Right.
tracking their kidney function and collecting samples.
Right.
This allows researchers to identify patterns and changes that may point to specific biomarkers.
They're like gold mines of information.
They really are.
Okay, so by looking at these detailed patient histories and linking them to molecular data, researchers can start to connect the dots between specific molecules and disease progression.
Exactly.
That's incredible.
And proteogenomics is already uncovering some promising candidates.
Okay, like what?
For instance, studies have shown that complement C1q,
Okay.
a protein typically involved in immune responses,
Right.
is elevated in the blood of CKD patients, suggesting its potential as a kidney-derived biomarker.
Okay.
And then there's uromodulin.
Yeah, we talked about that earlier.
We did.
In the context of ARPKD.
Yeah.
Is it also relevant to CKD more broadly?
Yes, it is. So uromodulin, also known as tan-horsefall protein, is produced by kidney cells.
Okay.
And intriguingly, its levels are actually lower in CKD patients.
Really?
And even lower in those with more advanced disease.
So both higher C1q and lower uromodulin could potentially serve as red flags for CKD.
Exactly. And there's a growing list of other proteins being investigated as potential CKD biomarkers,
Oh, wow.
each offering a different piece of the puzzle.
You mentioned cystatin C,
Yeah.
a trace protein, KIM1, NGAL, FABP, and even ADMA. It's amazing to see how proteogenomics is expanding our toolkit for CKD diagnosis and management.
It really is.
It all seems quite complex, though.
It can be.
What are some of the challenges that researchers face in this field?
Well, one of the biggest hurdles is simply the sheer volume of data generated.
I can imagine.
Integrating data from different omics platforms,
Yeah.
genomics, transcriptomics, proteomics. It's like trying to solve a massive multidimensional puzzle.
It must require some serious computational power and
Right.
bioinformatics expertise.
You're absolutely right. We need powerful computers, sophisticated algorithms, and skilled bioinformaticians
Yeah.
to analyze and interpret all this data.
And then there's the issue of variability in data quality, especially in proteomics.
Yeah.
What do you mean by variability?
So different labs might use slightly different methods
Okay.
for sample preparation or analysis,
Yeah.
which can introduce variations in the results.
Right.
This makes it challenging to compare data across different studies.
So standardization seems crucial here.
It is.
What are some of the challenges in achieving that?
One of the challenges is the lack of widely adopted standardized protocols.
Yeah.
However, the good news is that there's a lot of work being done to develop standardized protocols and software tools
That's great.
for proteogenomic analysis.
I imagine this kind of cutting-edge research doesn't come cheap either.
You're right. High-throughput sequencing and mass spectrometry can be expensive,
Yeah.
especially for large-scale studies.
Right.
But thankfully, the cost of these technologies is gradually decreasing as they become more widely adopted.
Oh, that's good.
Yeah.
So despite these challenges, the future of proteogenomics in nephrology seems incredibly bright.
It really is. Imagine a future where we can detect kidney disease at its earliest stages,
Oh, wow.
even before symptoms appear,
Yeah.
using a simple blood or urine test.
Early detection is key.
It is.
What else can you envision for the future of this field?
What if we could predict how individual patients will respond to different treatments,
Wow.
avoiding trial and error and potential side effects?
Yeah, that would be amazing.
Right.
How far away are we from seeing that in practice?
It's difficult to give an exact timeline, but the progress we're seeing is encouraging.
Okay.
Ultimately, the goal is to develop entirely new therapies that target the specific molecular pathways disrupted in kidney diseases,
Okay.
leading to more effective and lasting treatments.
Wow. It's like we're entering a new era of precision medicine for kidney health.
Yeah, that's a great way to put it. And it's not just about CKD.
Right.
Proteogenomics holds promise for a wide range of kidney diseases,
Yeah.
from genetic disorders to those caused by infections or environmental toxins.
So it sounds like this field is moving at a rapid pace.
It is.
What are some of the key takeaways you want our listeners to remember?
I'd say the biggest takeaway is that proteogenomics offers a powerful new lens for understanding kidney diseases at the molecular level.
Okay.
By integrating genomics, transcriptomics, and proteomics, we can unravel the complexities of these diseases and develop more precise diagnostic tools, targeted therapies, and personalized treatment strategies.
So it's about moving away from the one-size-fits-all approach.
Exactly.
And towards treatments that are tailored to the individual.
Precisely. We're moving towards a future where we can identify those at highest risk,
Yeah.
intervene earlier, and select the most effective treatments based on a patient's unique molecular profile.
That's incredible.
Exciting.
It is exciting.
Yeah.
This has been an incredible deep dive into the world of proteogenomics.
Hmm.
Before we wrap up,
Yeah.
could you leave our listeners with a final thought, some things to ponder as they continue their own exploration of this fascinating field?
Absolutely. Imagine it like this. Every day, scientists are uncovering new secrets about your kidneys, revealing how they work at the most intricate level. This research is moving fast, and new discoveries are being made all the time. So stay curious, keep learning, and be an advocate for your own health.
Right.
Proteogenomics has the potential to change lives, including yours.
That's a great message to end on. We've only scratched the surface of this fascinating field today.
Yeah.
We encourage you to explore this topic further and let us know what excites you most about the potential of proteogenomics. Thanks for joining us.
Thank you.
Okay. It really feels like we're on the cusp of something incredible in nephrology, with all these advancements in personalized medicine.
Yeah, it's a truly transformative time.
It's exciting.
Yeah.
Okay, so we've talked about ccRCC and how proteogenomics is helping us understand this complex cancer and personalized treatment approaches. Now let's turn our attention to ARPKD,
Okay.
which I know is a genetic disorder that affects the kidneys.
Right.
Can you give us a quick overview of ARPKD?
Sure. ARPKD stands for Autosomal Recessive Polycystic Kidney Disease.
Okay.
It's caused by mutations in the PKHD1 gene, which is essential for normal kidney development and function.
So if this gene is faulty, what happens in the kidneys?
Essentially, the renal tubule epithelium, the cells that line the inside of the kidney tubules, start growing abnormally.
Okay.
This uncontrolled growth leads to the formation of cysts,
Right.
those fluid-filled sacs that we discussed earlier.
Yeah.
These cysts disrupt the kidney's normal filtering process, leading to a whole host of problems.
And I recall reading that proteogenomics is playing a key role in helping us understand the molecular mechanisms behind ARPKD.
Right.
Can you expand on that?
Absolutely.
Yeah.
One of the most powerful applications of proteogenomics is its ability to identify changes in the expression of proteins that are encoded by the faulty PKHD1 gene.
So we can actually see how those genetic mutations in PKHD1 directly translate into changes in protein production.
That's right. This allows researchers to understand how those alterations in protein production contribute to the development and progression of the disease.
Okay.
It's like tracing the path from the genetic blueprint to the protein workers to the disease manifestation itself.
This knowledge is crucial for developing effective treatments. Yeah.
It's absolutely essential. By understanding these molecular mechanisms, we can start to identify potential therapeutic targets.
Can you give us some examples of the therapeutic targets that researchers are exploring in ARPKD?
Sure, one promising avenue of research is focusing on blocking epithelial growth factors.
Okay.
Remember how we talked about the abnormal cell proliferation in ARPKD?
Right.
Well, these growth factors are key players in driving that uncontrolled growth.
So by targeting these growth factors, we could potentially slow down or even prevent cyst formation.
That's the idea. There's also some very exciting research looking at a protein called Polycystin 1,
Okay.
which is encoded by the PKHD1 gene. This protein seems to play a crucial role in maintaining the structural integrity of the kidney tubules.
So if we can find ways to boost the function of polycystin 1 or even compensate for its deficiency, we might be able to prevent or slow down cyst development.
Right.
That's really promising.
It is, and there's another area where proteogenomics is proving incredibly useful, the search for non-invasive diagnostic tools.
Okay.
Remember those extracellular vesicles we discussed earlier? Those tiny delivery trucks sent out by cells?
They were fascinating.
Well, it turns out that when cells are stressed, like in ARPKD, they release different types or amounts of these vesicles.
So by analyzing these vesicles in a patient's urine, we could potentially detect ARPKD early on or track disease progression without needing invasive procedures.
Exactly. It's a much less invasive way to get a glimpse into what's happening at the molecular level in the kidneys.
Okay.
This could have a major impact on how we diagnose and manage ARPKD.
This is all so impressive. Now, I'd like to shift gears a bit and talk about the broader implications of proteogenomics for kidney disease research.
Okay.
How is it influencing the search for biomarkers, those molecular clues that can tell us about disease presence or progression?
Biomarkers are becoming increasingly important in the field of nephrology.
Okay.
They have the potential to revolutionize how we diagnose and manage a wide range of kidney diseases.
And how is proteogenomics contributing to this biomarker revolution?
It's allowing us to look for these biomarkers in a much more comprehensive and precise way.
Okay.
By analyzing the proteins present in blood urine or even kidney tissue, we can identify specific proteins that are either elevated or reduced in patients with kidney disease.
So it's like having a molecular magnifying glass that allows us to see these subtle changes in protein levels that might signal a problem in the kidneys.
That's a great analogy, and these biomarkers can serve as early warning systems, helping us detect kidney disease before symptoms even appear.
Right.
This is particularly important for conditions like chronic kidney disease, CKD, which often doesn't show symptoms until it's quite advanced.
Early detection is key for so many diseases.
It is.
If we can catch CKD early, we can intervene earlier and potentially slow down its progression, right?
Absolutely. And proteogenomics is already uncovering some very promising biomarker candidates for CKD.
Like what?
For instance, studies have shown that complement C1 key, a protein involved in the immune system, is elevated in the blood of CKD patients.
So that could potentially be a red flag for CKD.
It could, and on the flip side, there's uromodulin, also known as tan horsefall protein.
Right, we talked about this one earlier in the context of ARPKD.
We did. It's interesting because its levels are actually lower in CKD patients, and even lower in those with more advanced disease.
So both higher levels of complement C1q and lower levels of uromodulin could serve as potential indicators of CKD.
Precisely. And there's a growing list of other proteins being investigated as potential biomarkers for CKD.
Okay.
Each of these proteins offers a different piece of the puzzle, helping us build a more complete picture of what's happening in the kidneys.
You mentioned cystatin C trace protein, KIM1, NGL, LFABP, and ADMA earlier.
Right.
It sounds like proteogenomics is really expanding our toolkit for CKD diagnosis and management.
It really is. And remember those large prospective CKD cohorts that we talked about earlier?
Yeah.
These studies are playing a crucial role in biomarker discovery by tracking patients over time and collecting valuable data.
So by combining the power of proteogenomics with these large-scale studies, we're getting a much deeper understanding of the molecular changes that occur in CKD.
We are.
This is all so exciting.
Yeah.
But I have to ask, what are some of the challenges that researchers face in this field?
One of the biggest challenges is the sheer volume of data generated.
Right.
Integrating data from different omics platforms, genomics, transcriptomics, proteomics is like trying to solve a massive, multidimensional jigsaw puzzle.
I can imagine you must require incredible computational power and specialized expertise.
You're absolutely right. We need high-performance computing, sophisticated algorithms, and skilled bioinformaticians to analyze and interpret all this data.
So it's not just about the technology itself, but also about having the right people and tools to make sense of all this information.
Right.
What other hurdles do researchers face?
Another challenge is ensuring data quality, especially in proteomics.
Okay.
Different labs might use slightly different methods for sample preparation or analysis, which can lead to variations in the results.
So standardization is key, right?
It is.
We need to make sure that everyone is using the same methods and protocols so that we can compare data across different studies.
Absolutely. The good news is that there's a lot of work being done to develop standardized protocols and software tools for proteogenomic analysis.
That's encouraging. And what about the cost?
Yeah.
I imagine all this cutting-edge research comes with a hefty price tag.
That's true. High-throughput sequencing in mass spectrometry can be expensive, especially for large-scale studies.
Right.
However, the cost of these technologies is steadily decreasing as they become more widely adopted.
Oh, that's good.
This is making proteogenomics more accessible to researchers around the world, which is fantastic news.
So despite these challenges, the future of proteogenomics in nephrology seems incredibly bright.
It really is.
What excites you most about the potential of this field?
What excites me most is the potential to transform how we approach kidney disease from diagnosis to treatment.
Okay.
Imagine a future where we can detect kidney disease at its earliest stages, even before symptoms appear, using a simple blood or urine test.
That would be incredible. Early detection is so crucial for so many diseases.
It is.
What else can you envision for the future of proteogenomics in nephrology?
What if we could predict how individual patients will respond to different treatments, avoiding the trial and error approach and sparing patients from unnecessary side effects?
That's a really powerful idea. How realistic is that?
It is.
How close are we to seeing that in practice?
It's difficult to give a precise timeline, but the progress we're making is very encouraging.
Okay.
Ultimately, the goal is to develop completely new therapies that target the specific molecular pathways that are disrupted in kidney diseases. This would lead to more effective and lasting treatments and potentially even cures for some kidney conditions.
It sounds like we're entering a new era of precision medicine for kidney health.
It absolutely is.
What an exciting time to be in this field.
It is.
And it's not just about CKD.
Right?
Proteogenomics holds promise for a wide range of kidney diseases, from genetic disorders like ARPKD to those caused by infections or environmental toxins.
Exactly.
So we've covered a lot of ground today exploring how proteogenomics is revolutionizing our understanding of kidney disease and paving the way for more personalized and effective treatments. As we wrap up, what are some of the key takeaways you want our listeners to remember about this fascinating field?
Yeah, it really does feel like we're on the cusp of a paradigm shift in how we approach kidney health.
Yeah, I think you're right.
It's very exciting.
It is a really exciting time for the field.
So we've talked about, the potential for early diagnosis, personalized treatments,
Yeah.
even the development of new therapies.
Mm-hmm.
But before we wrap up this deep dive,
I want to make sure that our listeners really take away some of the key points about proteogenomics.
Okay.
What are the most important things you want them to remember?
I think the most important point is that proteogenomics offers a completely new way of understanding kidney diseases.
Okay.
At the most fundamental level, the level of molecules.
Mm-hmm.
By integrating genomics, transcriptomics, and proteomics, we can start to untangle the complexity of these diseases
Right.
and develop more precise diagnostic tools, targeted therapies, and personalized treatment strategies.
So it's really about moving away from that one-size-fits-all approach to healthcare
Exactly.
and toward a future where treatments are tailored to the needs of each patient.
Precisely. We're moving toward a future where we can identify individuals who are at the highest risk for developing kidney disease,
Yeah.
intervene much earlier in the disease process, and select the most effective treatments based on a patient's unique molecular profile.
This has been an incredible journey into the world of proteogenomics.
It has.
I think our listeners are as inspired as I am. But before we say goodbye, I would love for you to leave everyone with a final thought.
Okay.
Something to really ponder
Sure.
as they continue to explore this field.
I think the most important thing for everyone to remember is that this field is rapidly evolving.
Yeah.
New discoveries are being made every day.
Mm-hmm.
Stay curious, stay engaged, and be an advocate for your own health.
Oh,
Proteogenomics has the potential to change lives, and that includes yours.
that's a great message to end on.
Thank you.
We've only scratched the surface of this amazing field today, but I hope we've inspired you to learn more. Thanks for joining us on this deep dive into the world of proteogenomics.
Thanks for having me.
This is Radio EBM.